enzymes and biological reactions. So, first you need to know the protein nature of enzymes. So, the term metabolism refers to all reactions of the body. Reactions occur in sequence called metabolic pathways. These include anabolic reactions, which are the building up of molecules, and catabolic reactions, with the breaking down of molecules, like digestion. Metabolic pathways are controlled by enzymes. The products of one enzyme-controlled reaction become reactants in the next. Enzymes are globular proteins that are catalysts, which are molecules that speed up chemical reactions. But the enzymes remain unchanged at the end of the reaction. Enzymes are called biological catalysts because they are made up of living cells. Enzymes and chemical catalysts share properties in the reaction they catalyze. So they speed up reactions, they are not used up, they aren't changed, they have a high turnover number, so they catalyze many reactions. Enzymes only catalyze reactions that are energetically favorable and would happen anyway. Without enzymes, reactions in cells would be too slow to be compatible with life. Enzymes are proteins with tertiary structure, and the protein chain folds into a spherical or globular shape with hydrophilic R groups on the outside of the molecule, making enzymes soluble. Each enzyme has a particular sequence of amino acids, and the elements in the R group determine the bonds that amino acids make with each other. These are hydrogen bonds, disulfide bridges, and ionic bonds, and they hold the enzyme molecules in its tertiary form. A small area with a specific 3D shape is the active site, and it gives the enzyme many of its properties. So, when they say small, it is quite small. So say this is the whole enzyme. The active site would only be like, say, this part of it, or maybe even smaller than that. So if it's like if you held your arms in a circle, it would just be the tip of one of your pinky fingers, just this part. So actually it would be quite smaller than that. It would just be more like that, wouldn't it? A very small part of the molecule. Chemical bonds vary in strength depending on the atom that they join and on their chemical environment. Hydrogen bonds are weak, but if there are many of them, they have a significant binding effect. Ionic and covalent bonds, such as disulfide bonds, are stronger than hydrogen bonds because hydrogen bonds occur between molecules, and ionic and covalent bonds occur in molecules. They're intramolecular, not inter. Sites of enzyme action. So enzymes, like other proteins, are made inside cells. There are 3 distinct sites where they act. Extracellular, which is outside of a cell, so some enzymes are secreted from cells by exocytosis and catalyze extracellular reactions, like amylase, which is made in the salivary glands, moves down to the salivary ducts to the mouth. Saprophytic fungi and bacteria secrete amylases, lipases, and proteases onto their food, which digest it, and then they absorb the products of digestion. There are 2 different types of intracellular. There's intracellular in solution. So intracellular enzymes act in solution inside cells, like enzymes that catalyze glucose breakdown in glycolysis, a stage of respiration, are in solution in the cytoplasm. Enzymes in solution in the stroma of the chloroplast catalyze the synthesis of glucose. I said that. They've done strange placement of commas. So there should have been an extra comma after the word respiration. So it would be said, enzymes that catalyze glucose breakdown in glycolysis, a stage of respiration, are in solution in the cytoplasm. And also enzymes in solution in the stroma of the chloroplast catalyze the synthesis of glucose. Intracellular, membrane-bound, there's another type of intracellular. Intracellular enzymes may be attached to membranes, for example on the cristae of mitochondria and on the grana of chloroplasts, where they transfer electrons and hydrogen ions in ATP formation. So, an active site is defined as the site on an enzyme with a particular 3D structure that permits binding with a substrate. An enzyme acts on its substrate, and with which it makes temporary bonds at the active site, forming an enzyme-substrate complex. When the reaction is complete, the products are released, leaving the enzyme unchanged and the active site ready to receive another substrate molecule. An enzyme substrate complex is defined as an intermediate structure formed during an enzyme catalyzed reaction in which the substrate and the enzyme bind temporarily, such that the substrates are close enough to react. So you have the lock and key model, which the theory behind that is the unique shape of the active site means that an enzyme can only catalyze one type of reaction. Other molecules with different shapes will not fit. Enzyme specificity means that an enzyme is specific for its substrate. This concept gave rise to the lock and key theory. The substrate is imagined fitting into the active site as a key fits into a lock, hence the name. The shapes of lock and key are specific to each other, therefore the mechanism of enzyme action can be explained using the lock and key theory. So I've drawn a diagram here. This is an example of the enzyme's active site usually aren't that big, but this is just easier to look at. So you'd have an enzyme, and that would be an active site, and this would be the substrate, like here. And because they're complementary in shape, the substrate fits into the active site of the enzyme, then they can combine to form an enzyme substrate complex, and then once it's been digested, for example, say this happens, that substrate has been split up into smaller parts, digested. Lysosome and the induced fit model. So observation that an enzyme's shape was altered by binding its substrate suggested that it was flexible and not rigid, as originally thought. An alternative model, the induced fit model, was proposed, suggesting the enzyme shape altered slightly to accommodate the substrate. So the enzyme's active site isn't exactly complementary to the substrate, but it's close enough that it can change its shape slightly to accommodate for the substrate. A good example of this is the enzyme lysosome, which is an antibacterial enzyme in human saliva, mucus, and tears. The active site of the enzyme is a groove, and sugars on a bacterial cell wall fit into the active site. When the sugars on the bacterial cell wall enter the active site of the lysosome, some of the amino acids at the active site move 7.5 nanometers, allowing the groove to close over the chain of sugars. This changes the shape of the active site of the lysosome. This strains the bonds holding the sugars together and lowers the activation energy needed to hydrolyze, so break, the bonds between the sugars. As a result, the cell wall is weakened, the bacteria absorb water by osmosis, and the bacterial cell bursts. Activation energy is the minimum energy that must be put into a chemical system for a reaction to occur. By lowering the activation energy, enzymes allow reactions to take place at the lower temperatures found in cells. So molecules must have enough kinetic energy to approach closely enough to react. The minimum energy required for molecules to react, breaking existing bonds in the reactants and making new ones, is the activation energy. One way of making chemicals react is to increase their kinetic energy, to make successful collisions between them more likely. So heat speeds up the reactions in the non-living systems, but in most living organisms, temperature above 40 degrees Celsius cause irreversible damage to proteins and they denature. So this shows the effect of enzyme on activation energy. So this would be the usual activation energy, but with an enzyme it's lowered. So instead of increasing the temperature, enzymes work by lowering the activation energy. When a substrate enters the active site of an enzyme, the shape of the enzyme molecule alters, allowing reactions to occur at lower temperatures than in the absence of enzymes. Most enzymes are inactive at 0 degrees Celsius, and if the temperature is raised, they become active again. Enzymes in most organisms denature above 40 degrees Celsius. Lowering the temperature does not restore their activity. Reactions need to overcome an energy barrier before they can get started. So you can compare this with a person pushing a boulder over the top of a hill. They have to exert energy to get the boulder over the top of the hill before it can roll down to the other side. I always find this metaphor quite funny because it always reminds me of Sisyphus, the Greek legend of a guy rolling a boulder up a mountain. Anyway, The cause of an enzyme-controlled reaction. My camera is slightly wonky. Whoa. Oh no, this is not going well for me. The progress of an enzyme-catalyzed reaction for a given concentration of substrate can be followed by measuring either the formation of the product or disappearance of substrate. The shape of the graph, this graph here, may be explained as follows. When the enzyme and substrate are first mixed together, there are many substrate molecules. Both enzyme and substrate molecules are in constant motion and collide. Substrate molecules bind to the active sites of the enzyme molecules. In a successful collision, substrate is broken down and the products are released. More active sites become filled with substrate molecules. The rate of reaction depends on the number of free active sites. If all other conditions are optimal, then there is excess substrate. The enzyme concentration is the limiting factor because it controls the rate of the reaction. So at first, you have plenty of substrates, so it's just how many enzymes you have that are able to digest those substrates. Because if you have an abundance of substrates, you only have a certain amount of enzymes and only a certain amount of active sites, so that is the limiting factor. As the reaction proceeds, there is less substrate and more product. The enzyme concentration is constant. The substrate concentration is the limiting factor then, because it controls the rate of reaction. So, as the substrate is being turned into the products, there's the same amount of enzymes, but there's not so much substrate, so they're not colliding so often, forming less enzyme-substrate complexes. Eventually, all the substrate has been used and no more product can be formed, so the line plateaus, just goes flat. So, the line goes through the origin because at 0 time, no reaction has happened yet. So it just goes there. That's where it starts. You may be asked to read the mass of product formed at a given time, or be asked to calculate the rate of formation or percentage change in product formation. So, rate of production is increase in mass over time, and percentage increase in mass is actual increase in mass times 100 over initial mass. The rate of reaction at any particular time is the gradient of a tangent at that point. So, if we were to find, say, the rate of reaction at the beginning, you'd draw a tangent there. Actually, it'll be better for, say, at point B here. You draw a tangent here, going with the, it just goes with whatever the gradient is at point B. Usually you're meant to use a ruler. And you just try and get it on point B with the least amount of the rest of the line touching your ruler. And then you draw your line there, and then from here you can see the change in y over change in x, and once you get the values of change in y over change in x, then that is the gradient of that point. So that would be the gradient of point B. The curve is the steepest at the start of the reaction. This gradient there is the initial rate of the reaction. So if they ask for the initial rate, just get the gradient of the beginning of the graph where it's the steepest. And it starts at the origin, which is 0, 0. Factors affecting enzyme action. Environmental factors such as temperature and pH change the three-dimensional structure of enzyme molecules. Bonds are broken and the configuration of the enzyme site is altered, changing the rate of reaction. The concentrations of enzyme and substrate also affect the rate of reaction by changing the number of enzyme-substrate complexes formed. In exam questions, they always like it when you say how many enzyme-substrate complexes are formed. So, if there are less or more, or if it's remained the same. The effect of temperature on the rate of enzyme action. So, increased temperature increases the kinetic energy of enzyme and substrate molecules, and they collide with enough energy more often. This increases the rate of reaction. In general, the rate of reaction doubles for each 10 degrees Celsius rise in temperature up to a particular temperature, about 40 degrees Celsius for most enzymes. Above this, temperature molecules have more kinetic energy, but the reaction rate goes down because the increasing vibration breaks the hydrogen bonds, which change the tertiary structure of the enzymes. This alters the shape of the active site, so the substrate no longer is able to bind to the active site, so the enzyme is therefore denatured, which is a permanent change in the structure. So denatured means an enzyme's active site is permanently distorted by the irreversible breaking of hydrogen bonds, preventing substrate binding and reducing the rate of reaction. When an enzyme molecule is denatured, its primary structure, the order of amino acids, is unaffected, but it loses higher levels of structure. At low temperatures, the enzyme is inactivated as the molecules have very low kinetic energy. However, the shape remains unchanged, and the enzyme will work again if the temperature is raised. Inactivation is the reversible reduction of enzyme activity at low temperature, as molecules have insufficient kinetic energy to form enzyme substrate complexes. So on this graph you can see between about 0 to 30 degrees temperature, it's inactive at 0, but it becomes active as it gains more kinetic energy. And at optimum temperature it's working its best, but past optimum temperature it has too much kinetic energy, the hydrogen bonds break, which alters the... I was just checking if tertiary was the right word. It is tertiary. So it alters the tertiary shape of the enzyme, which alters the shape of the active site, so no enzyme substrate complexes can be made, so the rate of reaction rapidly drops. So the effect of pH on the rate of enzyme action. Most enzymes have an optimum pH at which the rate of reaction is highest. Small pH changes around the optimum causes small reversible changes in enzyme structure and reduces its activity, but extremes in pH denatures enzymes extremes of pH permanently denature enzymes. The charges on the amino acids side chains of the enzyme's active site are affected by hydrogen or hydroxyl ions, so H plus or OH minus. At low pH, so acids, excess H plus ions are attracted to the negative charges and neutralize them. At high pH, excess OH minus ions neutralize the positive charges. This disrupts the ionic and hydrogen bonds, maintaining the shape of the active site. As a result, the shape changes, denaturing the enzyme. This means no enzyme substrate complexes form and enzyme activity is lost. A small change in pH level is just about just one level above or below optimum. So, it's less than 1 pH value from optimum. So instead of being, say, pH 7, it's pH 7.5. But above 1 pH level of change, that is a extreme of pH changing. When enzymes are denatured by high temperatures or extremes of pH, their hydrogen and ionic bonds are broken, as I said before. Some chemicals that denature enzymes can also break disulfide bonds. To prevent pH fluctuations in a reaction, a buffer must be added, so buffers maintain a constant pH even when the products of the reaction are acid or alkaline. Buffers may be thought as absorbing extra hydrogen or hydroxyl ions. So there's a little diagram here that helps you understand. So acidity is in pink and red. So acidity has extra H plus ions, and alkaline have excess OH minus ions, and these excess H plus ions and OH minus ions break the bonds within the enzyme. So substrate concentration. The rate of reaction of an enzyme-catalyzed reaction varies with changes in substrate concentration. If the enzyme concentration is constant, the rate of reaction increases as the substrate concentration increases. At low substrate concentrations, the enzyme molecules have only a few substrate molecules to collide with, so the active sites are not working at full capacity. With more substrates, more active sites are filled. The concentration of substrate is controlling the rate of reaction, and so it is a limiting factor. As even more substrate is added at a critical concentration, all the active sites become occupied, and the rate of reaction is at its maximum. When all the active sites are full, the enzyme is saturated. Even if more substrate is added, reactions cannot be catalyzed any faster, so the plane plateaus. The substrate concentration is no longer controlling the rate of reaction, so it is no longer a limiting factor. Some other factor controls the rate of reaction, such as temperature. I should say factors. Some other factors control the rate of action, such as temperature reaction. A factor becomes limiting when an increase in its value causes an increase in the rate of reaction. So here there's a diagram. Substrate concentration increases, rate of reaction increases, so it's the limiting factor here. But then substrate concentration is no longer the limiting factor. There's not enough enzymes for the amount of substrate, so enzymes become the limiting factor, so the line plateaus. There's also a diagram to show the effect of high and low substrate concentration. So low substrate concentration, there's empty active sites, and it's only limited number of substrate molecules to fill active sites. So that's substrates being the limiting factor. Here, high substrate concentrations, there's excess substrate molecules unable to find any free active sites, so that's what is limiting the rate of reaction. Enzyme concentration. Once a product leaves the active site, the enzyme molecule can be reused, so only a low enzyme concentration is needed to catalyze a large number of reactions. The number of substrate molecules that one enzyme can turn into products in a given time is the turnover number. One of the fastest acting enzymes known is catalase, with a turnover number of 40 million molecules per second. It breaks down the highly toxic waste hydrogen peroxide. As the enzyme concentration increases, there are more active sites available, and therefore the rate of reaction increases. If temperature and pH are optimal, and there is an excess of substrate, the rate of reaction is directly proportional to the enzyme concentration. Enzyme inhibition is the decrease in rate of an enzyme-controlled reaction by another molecule, an inhibitor. An inhibitor combines with an enzyme, prevents it from forming an enzyme-substrate complex. So, inhibitor, a molecule or ion that reduces the rate of an enzyme-controlled reaction. Competitive inhibitors have a similar shape to the genuine substrate, and have a molecular shape complementary to the active site and similar to that of the substrate, so that they compete for the active site and prevent the substrate from binding. For example, in the mitochondrial matrix, a reaction, the Krebs cycle, is catalyzed by the enzyme succinic dehydrogenase. Malonic acid has a similar shape to succinic acid, and so they compete for the active site of succinic dehydrogenase. Increasing the concentration of the substrate, succinic acid reduces the effect of the inhibitor, because the more substrate molecules present, the greater their chance of binding to active sites, leaving fewer available for the inhibitor. But if the inhibitor concentration increases, it binds to more active sites, and so the rate of reaction is slower. As the concentration of competitive inhibitors increase, the rate of reaction decreases. However much inhibitor is present, the massive product formed ends up the same, because the inhibitor can bind to the active site and unbind, so in other words, inhibition is reversible. So eventually, all substrate is broken down. The competitive inhibitor is not permanently bound to the active site, so when it leaves, another molecule can take its place. This could be substrate or inhibitor, depending on their relative concentrations. So this is competitive inhibition. So you have a substrate molecule occupying the active site, and that's an enzyme-substrate complex. But then you have an inhibitor molecule occupying the same shape of active site, which means less substrate molecules are able to occupy the active site because the inhibitor is occupying those active sites. So that would be called an enzyme inhibitor complex, and this would be an enzyme substrate complex. There are also non-competitive inhibitors. So non-competitive inhibitors bind to the enzyme at the allosteric site, which is a site other than the active site, so they don't compete with the substrate. However, they do affect the bonds within the enzyme molecule and alter its overall shape, including that of the active site. So the substrate cannot bind with the active site and no enzyme-substrate complexes form. As the inhibitor concentration increases, more enzyme molecules are denatured, and so the rate of reaction and final mass of product decrease. Examples of non-competitive inhibitors include heavy metal ions like lead or arsenic. Some non-competitive inhibitors bind reversibly and some irreversibly. So non-competitive inhibition is defined as reduction of the rate of an enzyme-controlled reaction by a molecule or ion that binds to the enzyme substrate somewhere other than the active site, altering the shape of the active site so that the substrate cannot bind. So the cyanide ion is an inhibitor of the respiratory enzyme cytochrome oxidase. It is unusual because although it is not a competitive inhibitor, it binds at the active site. So this is to show non-competitive inhibition. So you have a substrate molecule occupying the active site, so this is what would usually happen. But if a non-competitive inhibitor binds to the allosteric site, it changes the shape of the active site so the substrate molecule can no longer bind to the active site. So, overall, end product inhibition occurs when a product of a series of reactions inhibits an enzyme that acts earlier in the series, so it slows down the whole sequence of reactions. That's a very important way of controlling cell metabolism. You can immobilize enzymes. So enzymes are called immobilized when they're fixed, bound, or trapped in an inert matrix. Inert just means unreactive, such as sodium alginate beads or cellulose microfibrils. Immobilized enzymes are defined as an enzyme molecule bound to an inert material over which the substrate molecules move. These can be packed into glass columns. Substrate is added to the top of the column. As it flows down, its molecules bind to the enzyme molecule's active sites, both on the bead surface and inside the beads, as the substrate molecules diffuse in. Once set up, the column can be used repeatedly. The enzyme is fixed and does not contaminate the products. The products are therefore easy to purify. Immobilized enzymes are used widely in industrial processes such as fermentation, but they can be readily recovered for reuse. Another good example of this would be production of lactose-reduced milk. So you have beads of immobilized lactase enzyme in beads, and then milk is poured over it. And there's like a little tap at the bottom that you can close so you can alter the flow. If there's a slower flow, it means there's a longer contact time between the immobilized enzymes and the substrates in the milk, which means that more enzyme structural complexes are formed, which means more lactose is broken down. The size of the beads does affect also the rate of reaction. So if you have smaller beads in the same amount of space, I'll just draw it here, it has an overall larger surface area for the substrate to move over, and diffusion distances are also shorter to get to the enzyme to the inside of the beads, whereas larger beads, diffusion distances are larger, and it just decreases the rate of enzyme substrates being formed because the surface area is overall smaller. So yeah, enzymes are widely used in industrial processes rather than in organic catalysts because they are more efficient and have the following advantages. They have a higher turnover number, they're very specific, and they are more economical because they work at lower temperatures. If a given volume of material is used to make large beads, there will be a similar total surface area than if the same volume had been used to make small beads. As a result, if small beads are made, the substrate molecules will have an easier access to enzyme molecules, and so they will produce a higher rate of reaction. Enzyme instability is one factor preventing the wider use of enzymes that are free in solution, Organic solvents, high temperatures and extremes of pH can all denature enzymes with a consequent loss of activity. Immobilizing enzymes with a polymer matrix makes them more stable because it creates a microenvironment allowing reactions to occur at high temperatures or more extreme pHs than normal. Trapping an enzyme molecule prevents the shape changes that denature its active site, so the enzyme can be used in a wider range of physical conditions than if it were free in solution. So I'd say the most important advantages of immobilized enzymes. I'll say them in order of most important. I have it written down here. Most important, enzymes are easily recovered for reuse, so they don't get wasted, and they're in a fixed position, so it's easy to just recover them. Products are not contaminated with the enzyme because they're fixed in place. They don't have the enzyme in the final product. A sequence of several columns can be used, so several enzymes with differing pH or temperature optima can be used in one process. Enzymes can be easily added or removed, giving greater control over the reaction, and increased stability and function over a wider range of temperature and pH than enzymes free in solution. So, uses of immobilized enzymes, you have lactose-free milk. An important industrial use of immobilized enzymes is in making lactose-reduced or lactose-free milk. The milk passed down the column containing immobilized lactase. The milk is passed down the column containing immobilized lactase. The lactose binds to its active site and is hydrolyzed into its components, alpha glucose and galactose. Biosensors, so a major use of immobilized enzymes is biosensors, which turn a chemical signal into an electrical signal. A biosensor is defined as a device that combines a biomolecule, such as an enzyme, with a transducer, to produce an electrical signal which measures the concentration of a chemical. Biosensors rapidly and accurately detect, identify, and measure even very low concentrations of important molecules. They work on the principle that enzymes are specific and are able to select one type of molecule from the mixture, even at very low concentrations. One particular use of a biosensor is in the detection of blood glucose. Immobilized on a selectively permeable membrane placed in a blood sample, binds glucose. This produces a small electric current detected by an electrode and read on the screen. Enzymes can also be immobilized onto test strips, where different strips may detect a variety of molecules. Testing strips with glucose oxidase immobilized onto them are used for detecting glucose in urine. There's also high fructose corn syrup manufacture. So HFCS is manufactured in a multi-step process from starch. It uses several immobilized enzymes requiring different physical conditions. So you have starch, and it's reacted with alpha amylase, which digests it into oligosaccharides, and then that's digested by glucoamylase, which turns it into glucose, and then glucoisomerase is used to turn it into fructose. And that's the whole topic. That's enzymes and biological reactions.